So let's do a quick overview of SARS-CoV-2. The coronaviruses are classified as alpha, beta, gamma, delta lineages, and SARS-CoV-2 is a beta coronavirus. Vaccine research focuses on the prototype Wuhan strain. And this was a strain that wasn't a strain as such because genetic sequence was determined in the second week of January 2020, and everyone took the genome sequence and used that to make vaccine candidates. There was no virus till much later. The focus is mostly on the spike protein. It's a major protein on the surface of the virus. It binds to the cell receptor, angiotensin converting enzyme 2, via something called the receptor binding domain, or RBD. So the focus of much of the vaccine development has been on the RBD, the idea being to get antibodies that block binding to the cell receptor and also neutralizing activity. The protein has two forms, spike, of pre and post fusion. Pre-fusion is found at the virion, and post-fusion is found in infected cells where it goes through a conformational change. So why is all this important? So we look at the left-hand panel at the bottom in B. This is the pre-fusion form, which sits perpendicular to the virus surface. The RBD in green on the surface is where all the action is. And if you look at the post-fusion form on the right, you can see there's a conformational change in the protein, and so essentially it's not as good an imaging as the pre-fusion form. The spike protein is about 1,300 amino acids in length. The amino half is called the S1 subunit, and the carboxy half is called the S2. And as you can see, the RBD is in green, is in the S1 domain. And there's other motifs there, we haven't really got time to talk about those. So in terms of vaccine imagings, you name the approach that's been tried. We have inactivated vaccines, which are inactivated with beta-progalactan or formula or both. We have the entire spike protein, either pre or post fusion. We have the S1 and S2 subunit, the S1 subunit receptor binding domain, spike protein plus other structural proteins, envelope membrane, nucleocapsid, or we have vaccines based on non-structural proteins or peptides, and this is to make T-cell based vaccines. So a lot of different types of imaging have been used. And in terms of platform technologies, again, you name it, it's been tried. Live attenuated viral, live attenuated bacterial vector, inactivated with or without one or two adjuvants, recombinant protein, messenger RNA, DNA, replicating bacterial vector, replicating viral vector, non-replicating viral vector, virus-like particles, and T-cell. And I can go through that quite quickly because all those candidates have actually been through evaluation, and we'll talk more about it as we go through it. So you name the technology and it's been applied. So how many COVID vaccine candidates there have been? As of this month, about 350 have been developed. It's an amazing number. Additional 50 to 70 in academic labs and small biotechs. And I think the most amazing figure to me is actually 180 of those have made it into clinical trials. And even the phase 1 clinical trial is going to set you back the best part of 20 million, if not more, to do it. And so that's the amount of money that's being thrown at doing COVID vaccine development. So from that, 36 authorized, I'll explain that in a minute, or licensed vaccines that have been found around the world, and they're used in 197 countries. 11 have what's called WHO emergency use listing, which means they're approved for use in multiple countries. The cost of these vaccines vary from $2 to $40 a dose, and the fastest development on record for a vaccine has been achieved of 326 days from discovery to authorization for Pfizer. And I'm not saying that's Rogers here, but actually it's an amazing record. Normally it takes between 9 and 20 years to get a vaccine through the whole pipeline from discovery to licensure. Here it's been done in 326 days. And of course the reason it got done was everybody wanted to work together to make a vaccine. It was a pandemic. And secondly, money wasn't an object, right? Money was just thrown everywhere in this field. And so that's why it went so fast. No shortcuts were taken. The clinical trials were crazy and brutal, I think it's fair to say. The whole process was. Everyone worked really very, very hard to do these studies and get everyone enrolled in the clinical trials, evaluate the data and get the data to the regulator. And I'm ad-libbing because I'm going over time, so I apologise. But one of the things about the clinical trials was that it was unusual. Normally, you get the data for clinical trial, then you report it every 6 weeks or something to the monitor or to the regulator for the company. In the case of Pfizer, we were reporting every night. And as I said, it was brutal. So you would do the clinical trial during the day. The QA team would come in and do the quality assurance. That data would then be updated to Pfizer, Moderna, etc. in the evening. By the time we get up in the morning, the queries have come in and they have to be addressed before 10 a.m. before the next volunteers come in. And that was happening 7 days a week. It was interesting. I think it's a good way of putting that. I'm sure Roger has similar stories from the side of the business. So where are we with COVID vaccines around the world? I said there were 36 vaccines approved or licensed, and this is just showing some of those. I've color-coded it to make it easy to follow. In red are the messenger RNA vaccines. The blue category next down are the adenovirus platforms using defective adenoviruses. The green ones are recombinant proteins, spike proteins with or without different adjuvants. And then we have one live attenuated vaccine, believe it or not. The Human Childhood Institute from Moscow developed a live attenuated vaccine based on codon deoptimization. That is to say, you have codons optimized for multiplication of the virus. So if you change the codon so it's not translated efficiently, you can actually make a vaccine out of that. And that approach was used by Jumkov. The next blue category covers inactivated vaccines made by a variety of different entities. And then in black, the Finley Research Institute in Cuba, they made a vaccine based on the RBD, chemically bound to tetrodotoxoides and adjuvants. Vector Institute, Novosibirsk in Russia, has something cryptically called SARS-CoV-2 peptides. I have to say I don't know what that is, but it's something that's actually been approved for use in Russia. And then finally, we have a DNA vaccine that's been approved in India. It's 3 doses, but it's given by injector, gas injector, so it's not a needle, so it's not going to be easy to use. Probably the best way of looking at these vaccines is to compare them in terms of efficacy. When we talk about vaccine efficacy, we're talking about a clinical trial being done blinded where you have a placebo group against the vaccine group. You give the placebo or the vaccine, you then follow, wait for SARS to take place, COVID to take place, and then you look at the number of cases in the control group versus those in the vaccine group. Subtract them from each other, and that becomes vaccine efficacy. And so the vaccine efficacy was 95% for Pfizer, 94.1% for Moderna. This is all during 2020 when we had the Wuhan prototype type disease around, and all the vaccines are compared at 1 month after the final dose of the vaccine. So all this data is comparable. So the best messenger RNA is the best. That's followed by recombinant protein, which are around about 90% efficacy. The inactivated vaccines are somewhere between 75-85%, depending on which type of inactivated vaccine it is. The defective adenoviruses are not as good. The Janssen is a single-dose vaccine, and the current efficacy that's been reported is 52.9%. I've got a question mark that it's been revised recently. The chimpanzee adeno for AstraZeneca is about 62%. Question mark there because no 2 trials are the same, and it's really hard to work out what the real efficacy is. Interestingly, Sputnik 5 that came out of Russia was the first one which used 2 different platforms, what we call heterologous prime boost, where the first dose was based on an Ad26 platform, but then the second dose is based on adenovirus type 5. And this is now part of the way it's moved forward, that if you use different platforms, that gives a much better immune response. And as you can see, 91.4% with 2-dose regimen. The DNA vaccine out of India only has 66% efficacy, and that's even after 3 doses, so it's not something that's going to set a bomb on fire. So you can see the messenger RNAs come out of the best platforms that we have at the moment. What about the United States? In the United States, we have a double process in approval of vaccines. The FDA licensed vaccines in the United States. The FDA makes a recommendation to its advisory committee called the Vaccines and Related Biological Products Advisory Committee, or VRBPAC. VRBPAC makes a recommendation back to the FDA based on the data. If it's approved, then it goes to the CDC Advisory Committee on Immunization Practices, and ACIP makes a decision on implementation. Why this becomes important is because if CDC decides to implement, then Medicare and insurance companies pay the cost. And of course, if they don't approve it, then the individuals pay. So when we go back to the start of the 21st century, we had anthrax attacks in 2003. And this was a wake-up call because we couldn't go through the normal process for what's called a BLA, a biologic license approval for drugs, vaccines, and diagnostics, because there just wasn't time in an emergency situation. And so the FDA under the Bush administration came up with something called the EUA, Emergency Use Authorization, where they can rapidly and temporarily approve the authorization of drugs, vaccines, and diagnostics to be used in an emergency scenario. But on the basis that once the emergency is over, then you have to go back and complete and get a full BLA approval. So let's look at the messenger RNA vaccines. In the US, that's Pfizer and Moderna. The technology I'm sure you're aware of is a lipid nanoparticle containing messenger RNA that gets taken up by endocytosis, and then you get the RNA leaving the endosome, escaping, translated, you make protein. So it's a bit like, in some ways, a live vaccine, but it keeps multiplying, but the multiplication is at the level of the RNA keep being translated to make more protein to be immunogenic, and it's very successful. And as you might imagine, we have different messenger RNA technologies, so the LNP and the RNA, and the parts of that are all proprietary. I'm sure Roger will not want to talk about that. But all the different forms of that are different. If we move to the adenoviruses, in this country we have the Janssen based on human adenovirus 26. And the idea here is adenoviruses cause common colds. Some of these are very rare, such as adenovirus 26. Not many of us have an immune response against it, and it's not a very virulent virus. And so you can use that as a vector to carry a foreign gene, such as the spike protein of SARS-CoV-2. It's made defective, so it only goes through one round of multiplication, and that's how you get your vaccine to work. The chimpanzee adeno has been up for approval for some time, but has not been so far in the United States. Most recently, we have a recombinant protein vaccine for Novavax. This is 2 doses of a recombinant spike protein with matrix 1 adjuvant, given the protein produced in insect cells, and it's 2 doses 21 days apart. The Novavax EUA was submitted back in January of 22 and was approved in June. You'll see in the next couple of slides. And the efficacy against Wuhan type virus in 2020 was around about 90% in a clinical trial involving about 10,000. So if we do a quick comparison of the vaccines approved in the United States as of this month, we have the Pfizer 2 doses 30 micrograms of messenger RNA. We have Moderna 2 doses 100 micrograms of messenger RNA. Janssen is 1 dose, the effective human adenovirus 26. I believe it's 5 by 10 to the 10 virus particles, but the FDA documentation doesn't explicitly say that. Novavax is the 5 micrograms of protein with 50 micrograms of adjuvant. EUA, first one was Pfizer, December the 11th, 2020, followed a week later by Moderna, followed by Janssen in February of 21, and then most recently Novavax in June of this year. I put an asterisk next to the June one because although it went through the recommendations, it's put on hold because Novavax changed their manufacturing process, and they were presented new data on June the 3rd just before the approval, and the FDA want to review that information before they decide whether or not to give it a EUA. The BLA for the Pfizer vaccine was some 9 months later, in August of 2021, and I've highlighted it as 200 million doses because normally to get to do a BLA, you normally get a clinical trial maybe involving 50,000 people with a couple of doses, so that's 100,000 doses. In this case, because of the EUA, 200 million doses of Pfizer vaccine have been used between the EUA and the BLA. So I guess you can say there's actually a lot of information on both the efficacy and safety of that product. The Moderna one eventually got its BLA for adults on January 21, 2022. Janssen's still waiting to get its BLA approval. Efficacy, I've said earlier, is 95% Pfizer, 94.1 Moderna, Janssen 52.9, and Novavax 90.4. In terms of the distribution of the vaccine in the US, I'm a little bit out of date, this is March's data, it's 58% Pfizer, 37% Moderna, and 5% Janssen. So it gives you a good idea of a snapshot of where we are in the United States at the current time. So let's ask the question, do vaccines prevent COVID? And yes, all the data based on vaccine efficacy studies done in clinical trials supports that the vaccines are very effective. The mechanism of protection of the vaccines is not known and likely differs because we're using different platform technologies, and so they're at least going to be qualitatively different. We don't have a correlate protection yet. Now, correlate protection is something we like in the vaccine business. It's a magic number that tells us whether the vaccine is going to work, and normally it's based on neutralizing antibodies. Such if you have a neutralizing antibody titer, say 1 in 50, we'll know you'll be protected against that particular disease. But we don't have a correlate protection for COVID at this present time. And it gets more and more complicated because of the virus evolving. I'll talk more about that in a minute. Many people believe that neutralizing antibodies are the correlate protection, but the evidence is not really that conclusive. There's no definitive neutralizing antibody titers that shows they will protect against the disease. And so we consider neutralizing antibodies actually to be a biomarker of protection. We can't do vaccine efficacy studies in 2022 because people have either been vaccinated, they've had clinical disease, or they're vaccine hesitant. So we can't do the traditional way of doing a clinical trial. And so we've now moved to immunobridging studies, where we're trying to identify biomarkers that can be used to translate from the vaccine efficacy to a correlate protection. And hopefully it will be antibodies or some cytokine, we'll have to wait and see. But there's a big push now to do immunobridging studies instead. Let's look at potential EUAs coming up in this country. I'm crystal ball gazing here. I think the Sanofi GSK vaccine will probably get its EUA in the second half of this year. This is a 2-dose regimen, 10 micrograms of protein, Wuhan, produced in insect cells together with GSK's AS03 adjuvant. Vaccine efficacy for this one's quite interesting. It was done during a time of Delta and Omicron, not during Wuhan, because it's more recent. And their data was 77% protection against symptomatic Delta infection and 58% efficacy against symptomatic Omicron. So you can see a Wuhan vaccine does not work as well against the more recent variants. Nonetheless, there was still 100% efficacy against severe COVID-19 hospitalization and 75% against moderate to severe disease. It hasn't gone to the FDA yet, but it has gone to the EMA for something called a conditional marketing approval, which is equivalent to the EUA we have in the United States and in Europe. So one would assume it's going to come here soon. The big problem we have with COVID is that the viruses keep mutating, and we keep getting more and more new variants appearing, and the challenge is how do you keep up with all these viruses that are changing. The WHO has a classification of genetic variants. They have an organization of labs around the world which are continually sequencing viruses from various countries. And they identify variants of interest from increasing reports of the same mutant being found in more than one geographic area, but there's no fundamental change in the virus. And then we have variants of concern, which either are mutants that have increased transmissibility, they cause more severe disease, the vaccine does not prevent clinical disease by the variant, or the virus is less neutralized by vaccine antibodies. So mutants have increased transmissibility. This is the major problem we have, that the virus keeps changing and it keeps becoming increasingly with each variant more and more transmissible. And I'll explain that in a minute. Fortunately, as the evolutionary biologists predicted, as the virus is evolving, the virus is becoming less virulent but becoming more transmissible. And so that's good news. All the vaccines that we use to date are based on the Wuhan strain, which does not prevent infection, but it does prevent severe clinical disease. And as we get more and more evolution now in the so-called Omicron variants that we have now, this is becoming a bigger problem. I'll talk more about that in a minute. And finally, of course, as you might want to expect, if you make a vaccine based on Wuhan, the antibodies will work best to get neutralized in Wuhan. As you make more variants, then the neutralization titers decrease. So we call this waning immunity. But the waning immunity we see in different ways. What does it mean? How do we categorize it? It's all very well saying it decreases, but what does that mean? And in particular, we have 8 different neutralization assays that are used around the world, and few of them use international standards so the data can be compared, which makes it hard to evaluate it. So yes, the immunity wanes, but of course, well, how long is it much to wane over time? When are we going to get to that low level where we're no longer protected? The CDC came up with its classification system in March 2021, and they have 4 categories: variants being monitored, variants of interest, variants of concern, and variants of high consequence. There's a whole load of variants being monitored, as you might expect: alpha, beta, gamma, delta, epsilon, eta, iota, kappa, 1.617, mu, and zeta, just for a few. And you can see it's a continual problem. We keep getting these new variants coming out all the time. We have no variants of interest in the United States, but we do have variants of concern around Omicron, and we have no variants of high consequence. So I'll say Omicron is a real problem. It keeps evolving incredibly fast. So we've gone from B, this is all in 2022, from B1.1529 to BA.1, BA1.1, BA2, BA2.12.1, BA3, BA4, BA5. And so you think we're in 6 months in the year, this virus is particularly a problem. It's Omicron. To put this in perspective, if you looked at 2020 on the left-hand side over here, we'd have Wuhan in 2020, we have the Alpha variant that started off as a wave in early 2021. We had the Delta that came in the second half of 2021. But look at Omicron. We've already had 3 waves in the first 6 months. We've had BA1.1, BA2, and BA2.12.1. and I'm going to show you data to say that BA.5 is now the one that's going to take off in the next few weeks. It's a big problem. These waves are coming quicker and quicker with the Omicron variants. So let's look at Omicron. Why is Omicron a problem? Because it was a quantum shift in the virus. If you look at the Delta variant on the left-hand side, we've got a few amino acid mutations in the spike protein, but hey, we can live with 4 or 5 or 6. But when it came to Omicron, we had over 30 amino acid mutations in the spike protein, and we had another 20 in different parts of the virus. And you can see the mutations everywhere. It really is a very different protein. It's a different beast, if that's the right way of putting it, in terms of trying to think about how you're going to have a vaccine against this virus. The phenotype of Omicron is less clinical disease, but far more transmissible. The key amino substitutions all over the place, particularly in the receptor binding domain. And so 15 of the 30 amino substitutions in the spike are in the RDD. And rather disappointingly, I think, for vaccines, there were 19 neutralizing monoclones prepared against the Wuhan vaccine, and 17 don't neutralize Omicron. Yet, Delta and Omicron compete with each other against the ACE2 receptor. And it really, I was very surprised at this. I did not predict this, but it shows I'm pretty stupid, because when I thought there were so many amino acid differences, they would not compete. But they do compete with the same receptor, despite all those amino acid substitutions. So where did it come from? The popular idea is an immune escape variant derived from an immunosuppressed individual. I find that hard to believe, and I'm probably wrong again, but I don't see how a virus can evolve from one person and can rapidly move and cover the world so quickly. More likely to me, and I have no evidence for this, is that it jumped into animals, mutated in animals, and jumped back into humans. And we have a number of examples of that happening with SARS-CoV-2 variants. Is it a different serotype? I think this is the big worry. We now move so far antigenically that we're now got a separate serotype of SARS-CoV-2 circulating. And then the question comes, are we going to get a Wuhan type variant, the son of Delta, for example, that came back, such that we have concurrently 2 different serotypes of SARS-CoV-2 circulating in the future? And I think that's the big worry as we move forward. This is one of my favorite slides because it's simplistic and makes it clear where the problem is with Omicron. All these different colors make the point. All these viruses rapidly are mutating to become very different viruses. So Omicron original to BA1, 2, 3, 4, 5 is genetic changes in the virus, transmissibility changes. This is really a big problem. These viruses rapidly evolving and really have a different phenotype. The next 3 slides just give an example of this. This is CDC data tracking variant proportions in the United States. And if you look on the left-hand side, this is June 11th. We're looking from March forward, and you can see we've moved from BA1.1 to BA.2 to BA212.1. So if this had been in 2021, we would only see a couple of waves, but we've already seen 3 waves in this country already, and we now got BA4 and BA5 coming up here. If you look at the percentage of the population, we're at 64% on June 11th with 12.1 and 14% with 2, and you can see the rest. Within 1 week, we're down to 56% for BA12.1, and BA5 is up to 23%. And you shift another week, and we're up to 36% is BA.5. So this is going to be the next wave coming through. It's moving incredibly quickly. This is not slow like we had between Alpha and Delta. We're now seeing great changes in the virus in a very short period of time. And it's going to make it really a great challenge to try and control a virus that moves so fast in terms of making vaccines. In terms of vaccine efficacy, all the vaccines we use in the US have lost significant amounts of efficacy against the Omicron variant, but they still remain effective in preventing severe disease, hospitalization, and death. And although we're seeing many more infections, we're seeing nowhere near the levels of hospitalization and severe disease that we had in 2021. Interestingly, when periods of last year, when we had Alpha and Delta, people who received a booster enjoyed stronger protection than those who received just the normal 2-dose primary series. But during Omicron now, that's all changed. Immunity is less strong, it falls faster. And this suggests that the ability of Omicron to evade protection by the vaccines is due to antigenic variation or potentially transmissibility. But it's hard to evaluate all these data because everybody's having their vaccine booster at different times. And trying to build that into analysis of the data is very hard. I'll talk more about that as we go along. Some interesting data come out of the UK and the CDC, which really demonstrate nicely the problems we have. Pfizer in the UK, Pfizer and Moderna, 2-dose vaccine regimen, gave only 10% vaccine efficacy against symptomatic COVID at 25 weeks post-vaccination. That's just a primary series. When you got the booster, the first one, vaccine efficacy went to 75%, but only lasted 4 weeks. 15 weeks post that booster, you're down to 25 to 40% efficacy. If you look in the US at hospitalization, the primary series of Pfizer and Moderna only gave 54% vaccine efficacy at 5 months post-vaccination. The booster pushed it up to 91%, but by 4 months, we're already down to 78% vaccine efficacy. So it's not very good. The problem, I think, is more on the italic at the bottom. People who had a natural Omicron infection at the start of 2022 are now having natural Omicron subvariant infections, suggesting that even if you had an immune response based on Omicron less than 6 months ago, you are not going to be protected against the subvariant. And that really is a big problem as we move forward if this proves to be correct. So the problem in many ways, I think, is a reproductive number. The R0 is something that we use to look at transmissibility of agents. And so R0 refers to how many people can one person infect. For SARS-CoV-1, the R0 is 3. The original Wuhan strain, it was 2 to 3. The Alpha variant was 4 to 5. Delta, I've said, is 5.08 because studies vary from 3.2 to 9. Omicron, the original one, was 8.2. And now the BA.2 is 8 to 12. So you can see we're steadily increasing the ability of SARS-CoV-2 variants to become more transmissible and infect more and more people all the time. And this obviously means that vaccination, herd immunity, is really going to be very difficult to achieve, because at that sort of R0 level, we're going to have to immunize probably 98% of the population and keep them immune. That's really going to be a very hard task to do. So if we look at vaccine-induced immunity, neutralizing antibodies appear to protect against clinical disease, and the quantity appears to predict protection from symptomatic disease. But neutralizing antibodies wane over time, and we consider other components of the immune response mediate protection or contribute to it, particularly with the variants. There's a big focus at the moment on cell-mediated immunity, and non-neutralizing antibodies looking at something called antibody-dependent cell cytotoxicity. So we're looking at other mechanisms that may be involved in protection beyond neutralizing antibodies. And with this in mind, how can neutralizing antibodies be used as part of protection when the different variants keep appearing? And where, as I said earlier, evaluation of neutralizing antibody titers has to be taken with great care. So as we move forward, the current vaccines have all been based on Wuhan to date. There's limited data available for efficacy of most of these approved licensed vaccines against the genetic variants, but data suggests that the Wuhan-based vaccine protects against severe disease, hospitalization, against all variants, maybe except for some of the Omicron. Until recently, we've seen convergent evolution of SARS-CoV-2, although Omicron is having the last. And I always think viruses are far more intelligent than humans. Every time we think we've got them beaten, they come along and do something different. One of the complexities of something we really need to sort out is the neutralization assays. We use 8 different assays. We have WHO standards available, but not everybody uses them. And then comes the question, when do you boost people for vaccination? How much space should there be for boosters? And that is a real problem. In this country, we've had 5 months recommended, now 4 months, and that's just not sustainable. People just don't want to keep getting vaccine boosters every 3 or 4 months. People have thought, well, I may be willing to take a booster every year, but that's not what we're seeing at the moment with the amount of COVID activity we're seeing. One of the interesting areas that's being looked at the moment is heterologous prime boost, so-called mix and match studies, as a way forward for vaccination. Sanofi just put a paper out recently as a preprint, so it's not peer-reviewed. And this used the Wuhan spike protein as the initial study to make their vaccine. They then tried heterologous prime boost in the middle of June, where they primed with 2 doses with the Pfizer messenger RNA vaccine, and then boosted with either the Pfizer messenger RNA, Sanofi original Wuhan vaccine, or the Sanofi beta variant. And they found the beta variant gave a superior neutralizing antibody response to the other 2 regimens, suggesting that this is the way forward. I think this tells us that we can actually use different platform technologies and get the improved immune response, and indeed other groups have shown the same data. I highlighted the boost from 3 to 7 months because this is only 400 people in this clinical trial, and they're having boosts from 3 to 7 months is a big difference in terms of looking at the amount of immunity in those people and knowing whether the vaccines work. I think that's one of the problems of, as I said earlier, doing immunobridging for clinical trials to make sure the data is actually understandable is very difficult today. Pfizer have just put a press release out on their bivalent boost. Here they did a phase 2/3 clinical trial with 30 micrograms and 60 microgram booster doses of a monovalent BA.1.1 against the bivalent Wuhan plus Omicron BA.1. And this is a study done in the over 56s, senior citizens maybe, and just over 1,000 people. And they looked at superiority. This is one of the big things in bridging. We want superiority. We don't want a new vaccine to be as good as what we have. We want it to be better. And they said define this as a ratio of neutralizing antibody geometric mean titers with a confidence interval greater than 1. And they found that the BA.1 vaccine gave super superior neutralization data with these GMRs of 2.23 and 3.15 respectively. And I never knew what super superiority was before then. I'm glad they published this because I never understood how a vaccine could be superior, another one could be super superior, but now I've been educated. The bivalent vaccine, interestingly, was not super superior. It was superior with the GMRs 1.56 and 1.97, but it wasn't as good as the Wuhan one. But the problem, of course, with this study is, not being disrespectful, was Omicron's moving so fast. It was done with the BA.1, went out to BA.5. And the question is, how do you keep up with the Omicron variants? Moderna did a similar study as well, using Omicron BA.1 together with Wuhan, and got very similar data again. But as you notice on the last bullet, for the neutralization titers for BA.4 and 5 were threefold lower than they were against BA.1. So this variants are moving very fast and are less neutralizable by earlier Omicron variants. One of the other areas that's actively being looked at is something called hybrid immunity, and this means people having natural infection and then getting vaccinated, or vaccinated and getting natural infection as a way of enhancing their immune responses. And this is, I think, has a number of questions about the moral way of looking at these things, of using natural infections as a way of vaccination. And the issues have become, how long the time do you leave between vaccination and natural infection to get this immunization approach? It doesn't appear to be that good, as evidenced by the fact that people who had an original Omicron infection at the start of 2022 are now getting a clinical disease from subvariants. One of the interesting trials that's going on at the moment, I have to say I'm biased because we're one of the sites involved in it, so I do have a conflict of interest. This is a NIAID study, which is called COVAIL, where they're comparing 15 different arms. It's an amazing study, where they've taken people who have had their 2 doses originally, then a booster, and are looking at the 4th booster, but doing it with natural infection, boosting with Pfizer, Moderna, Novavax, or starting with Pfizer, Moderna, Novavax, then boosted with natural infection, and then complicating it by times by 3 by using the Wuhan strain, the Omicron strain, or bivalent. And so it's a huge trial, and I think this is going to give a lot of data in understanding how to proceed, because they're looking at innate cellular human immune responses to understand what's the best approach. But this heterologous prime boost and this approach in this trial, I think, can actually be very educational. Hopefully it will get completed next month. So FDA, VRBPAC met yesterday, and they were trying to decide, what are we going to do in the fall? What vaccines do we recommend? Do we recommend Wuhan, or should we change to Omicron? And the vote was 90-2 to recommend a bivalent booster vaccines that target Omicron variants in the fall. However, the panel didn't make a recommendation on which Omicron subvariant should be included in the vaccine, and Roger's going to talk more about that one in a minute. Other things are going on as well in the background. A large amount of money has been distributed to make pan-SARS-CoV vaccines. As I said earlier, we've got these alpha, beta, gamma, delta lineages, and so SARS-CoV is beta coronaviruses, and the idea is to have a vaccine that will work against SARS-CoV-1, SARS-CoV-2, the variants, and Middle Eastern respiratory syndrome. And though that funding's all out, and we'll have to wait and see how it goes. Not in the public domain, I'm afraid, but it looks quite promising. So the big picture where we are at the moment in terms of pandemic preparedness is we've had our first generation COVID vaccines based on Wuhan. We've got our second generation vaccines coming along based on variants. Do we need them? Yes. The beta coronavirus vaccines are now been, the funding's out there. Similarly, the funding is also out there for the pan-coronavirus vaccine to work against alpha, beta, gamma, and delta lineages. But of course, now this is in the public domain yet. And there's this large pandemic preparedness program by NIAID, SEPI, WHO, European Union, and many other entities. The NH1 has decided to focus on prototype strains. They've identified disease X as coming, the next one from one of these families: paramyxos, flavies, togas, picornas, philos, arenas, pneumos, peribunias, niros, and hanters. And the idea is if you can actually characterize and make a vaccine against a prototype strain, you can then do what we call plug and play, where you take another virus from that family and just place it between the prototype. That's how that works. We're in an era of pandemic exhaustion now. Everybody has to have these frequent boosters. How do we persuade people to take vaccines? Compliance. Who will follow the vaccine recommendations? How do we develop a vaccine that's going to last more than 4 months? And the Holy Grail, according to the head of Pfizer, is a vaccine that will work for 12 months. And I think that's where we are. That's the Holy Grail. We know we need that at the moment. Fewer and fewer people want to wear face masks. People have been vaccinated, are far less to get booster doses. And I think it's interesting. At the end of May, 2/3 of Americans have been fully vaccinated with a 2-dose regimen. Less than 1/3 have received a booster, and less than 5% have taken the second booster that's been recommended. We're in a time of exhaustion. How do we move forward? I want to finish and talk about a WHO report. Nothing changes. WHO had an independent panel for pandemic preparedness and response that did an evaluation of where we were in May of this year. They said not much has changed. Political leadership, financing, and surveillance systems haven't really changed. There is still vaccine equity between different countries. Indeed, as of May the 18th, only 15.9% of the 1.2 billion people in Africa have actually received the vaccine. The more worrying one, I think, is the last bullet. Where about 11.9 billion people now have received coronavirus vaccines around the world. If we look at the week of June 27th last year, 325 million doses of vaccine were distributed around the world in that 1 week. If you look at the May 22nd week of 2022, we're down to 38 million doses of vaccine distributed around the world. We have a big problem. People are fed up taking vaccines. Everybody is exhausted and just wants to get the pandemic out of the way. How do we actually solve this problem? And to me, this is going to be the big problem moving forward. How do we persuade people to take vaccines? Thank you very much.
So my disclaimer here is these are my opinions. Even though I'm at Pfizer and even though I've been working on the vaccine for the last 2.5 years, a lot of what I'm presenting here is my observations based on that and based on talking to people like Alan and interacting in the industry. One of the things that I want to stress here is this is the third, and I guess we've been doing this every year now in June, talking about this. And we started out talking about the vaccines before they were vaccines, when we were running clinical trials. And so as a result, we in a very short period of time, we've seen an extraordinary growth from what I would call, the infancy of this to where we're sort of in that adolescence. And actually a lot of what Alan talked about is a lot related to adolescence in terms of, where are we going? It's pretty confusing, it's pretty chaotic, and there are an awful lot of things that we have to learn yet. So the content for this is where we've been and where we are going. And I'm going to spend a fair amount of time, at least in the beginning, trying to differentiate a little bit about some of this new variant approach that's being handled. And I'm going to focus on Pfizer more than I am on the other companies because I'm more familiar with that. It doesn't add any clarity on the direction we need to go unless there's better collaboration between Pfizer and other companies, between the governments and the companies themselves. And that's a point I'm going to stress several times during this discussion. So there are some statistics, very simple statistics. In the first presentation I gave in 2020, I showed a lot of different charts from WHO and from other sources, Johns Hopkins, where you were seeing these exponential growth in the disease manifesting itself, the number of doses that were being generated, blah, blah, blah. Well, we're at sort of a stasis now, and Alan alluded to this. I'll also talk a little bit about the development of the mRNA vaccine and where we got to and where we are right now. And then I'll talk a little bit about addressing the variants. So the global data to date, and this is as of the 21st of June, 2022. I think Alan had 11.5 billion doses. I have 11.9. I think the difference fairly insignificant. I think the thing to point out here are the number of deaths. Confirmed cases, I think, is a little bit nebulous, because at this point, we don't know how many confirmed cases there are. There's so many people who test themselves at home, get COVID, don't report it, so you can't really determine that. But the deaths truly are significant, and I think that's something that we have to keep in mind. Severe disease and death is one of the things that we focused on from the very beginning. We weren't trying to get rid of the disease, although we certainly hoped that we could prevent it from occurring. Our real hope was that we could reduce the hospitalizations and deaths. So I'm going to show two timelines in succession, and I don't expect you to look at these in any detail, but I want to highlight a couple things here because they're kind of important. In the beginning of 2020, there was a huge amount of work done to understand the constructs related to which direction we were going to go with the Wuhan variant. We called it the wild type variant. Pfizer started out with several constructs. We actually had 11 to start with. We put 4 in the clinic. Of those 4, we ended up with 1. And when we ended up and selected the one we wanted, which was on June 21st of that year, the development machine started, and it started in earnest. I should say the development machine actually started even before that, because we were doing things even before we knew which construct we were going to work with. Constructs were very similar. They were all encapsulated. And so it wasn't very difficult to make some decisions early on about what we needed to do. What was different, and what I will sort of iterate here, and I iterated in previous talks, is the parallel versus sequential development that goes on. When Alan alluded to the 9 years it takes normally for a conventional vaccine to be developed and approved, it's because we do a lot of sequential work. We use science to make decisions about the next science we're going to do, and we continue to do that until we have a record of what we believe is appropriate product. In this case, we did science on top of science, and then we went back and reevaluated. So we were developing the drug product simultaneously while we were developing the drug substance, simultaneously while we were also manufacturing in several different nodes in different locations, and then comparing the data and determining what the best direction was. A little bit hazardous to do that because you spend a lot of money up front, but considering the pandemic, it was the right thing to do. Could you do it for everything? And this may allude to the question that you had earlier. I don't think you can, and I'll tell you why when we get to this question and answer part. The other thing I'd like to highlight here is that these VRPECs come fast and furious. So we had the VRPEC in November or December, but it actually was originally scheduled in November. And the FDA and the CDC wanted all this data and information immediately. And as Alan said, we were daily going over data, clinical data with them, so that they had answers or conjectures about what we were going to be ultimately presenting. But we were talking about 44,000 subjects worth of data over a very compressed 3-month timeline. And so in order to draw conclusions from that in a day-by-day succession is not the best way to do science. And that's one of the reasons this got pushed back to December from mid-November, because there was no way that we could all come to the same conclusion. And we wanted the VRPEC to be successful and that a good decision was made about whether to proceed, which is what happened in that case. That was driven largely by conversations between Peter Marks and Pfizer people like myself, because we had to do that. We had no choice. The CDC was not involved originally, and the 21 members of the panel were not involved in that discussion. They're supposed to be independent, so we had to make that decision in advance. Interestingly, once this occurred and once the decision was made, the approvals were very quick. And these are for the emergency use authorization, as Alan pointed out, or conditional approval in Europe. I mentioned the constructs, okay. The second timeline is 2021 and 2022, into 2022. So the 2021 from the beginning of the year to the end, and then the early part of 2022. Again, you see a VRPEC here in June 15th. That was on the pediatric doses. That we didn't even talk about that. Alan didn't mention the pediatric piece because the data there is confusing. And so confusing that we ended up going in a bit of circle trying to figure out what the outcomes were going to be. Again, limited number of patients, limited number of opportunities, a very tight time frame for boosts. So we were talking about 1, 2, 3, or 4 possibly injections in pediatric patients. That's an awful lot to expect for children in a short period of time. But again, there was this urgency, and the urgency was propelling us to move in that direction. The other thing to notice here, up to the end of 2021, we had done a lot of optimization with the process. And I'm a manufacturing guy. I'm a CMC guy. So I was involved with my team trying to get as much of an expanded footprint for manufacturing around the world as possible, so we could deliver any kind of vaccine with the platform we had. One of the nice things about mRNA is that the platform is relatively straightforward to work with. If you have the right constructs, you can actually build your mRNA platform drug substance, encapsulate it relatively easily, and have a drug product relatively quickly. But in order to do it, all the sites have to be operating together. And right now, the number of manufacturing nodes that Pfizer has is 37 different sites around the world working just on this. In 2022, Alan alluded to the Omicron, the emergence of Omicron, and what we should do with the variants. I can tell you that when we started with the Omicron variants, we were looking at, as they were emerging, we were looking at a number of constructs for the initial Omicron. And then immediately thereafter, as these new versions started to appear, trying to figure out whether or not we could use that as a baseline in our clinical trials and then supplement that later. But it takes time to do this. You cannot do it immediately. You have to get data, understand what the data says, and then move forward. And I think that's the conundrum and the challenge that Alan was alluding to. So a summary of the development itself and what was good about it and what actually worked was that we called it the Lightspeed designation, rapid parallel development. The platform, as I said, lends itself to being very, very amenable to rapid development. And we could transfer processes and analytics very quickly around the world. And those were real key factors that allowed us to move as quickly as we did. There was also tremendous collaboration in that first year. I've been in the industry 41 years, and I've never seen the amount of collaboration between companies, among regulators, among government officials to try to move very quickly. It was kind of nice to see, actually. Money was no object, and everybody was playing in the sandbox nicely. I think over the last year or so, I've seen a little bit of a change. As we've gotten commercial licenses, we now have ownership in a different way than we had when we got the vaccines approved under emergency use, which means we now have the obligation to determine what we're charging for these vaccines, where we're going to have them administered, who's going to buy them. And that changes the dynamic a bit, and I think it could change it in a negative way unless we're careful. So as we look at other pandemics in the future, it's something to think about because it may not be the right direction to go in. One of the other things that I thought was really, really good was the regulatory authorities were with us the whole way along, the entire way. We did not have as much collaboration with regulatory authorities on any other project I've worked on as we did with this. They were willing to accept, based on a limited amount of data with some measure of risk, as long as we were able to commit to provide that data to substantiate whatever that risk showed. And we did that, and that's one of the reasons it became a reliable approach, especially in that first year. And then there was alignment among the authorities themselves. I remember a conversation I had with the head of the Brazilian agency, Anvisa, where he asked me, he said, Roger, I know you guys have gotten good results and approvals or authorizations from the US and Europe. We would like to get the vaccine in Brazil. Our laws are prohibitive and don't allow us to get it in the way that you have been able to get it in the US and Europe, but I can use a mutually reliant or recognized approach by saying that the US and Europe have approved it, and if we have all that data, we can have it approved and distributed within a matter of weeks. That's pretty remarkable because they were willing to essentially obviate their statutory expectations in order to do that, which is something that I have not seen in the industry ever. So again, pandemic allowed us to move in that direction. It also made it much more attractive for us to be able to talk to these regulatory authorities en masse, not just one at a time, but 3, 4, 5, 6 regulatory authorities at once, because they were all willing to move in that same direction. Some of the things we learned that are important, probably I would say these are pretty obvious to many people. Don't let the bureaucracy get in the way. We had a collaborative team of people who had worked together for a number of years. So the experience level and the willingness to put egos aside was extraordinarily good in our case. And that's one of the reasons we were able to move. We were also connecting with our brethren around the world in other countries and other companies. We were not afraid to stick our neck out to say, hey, we've got this issue. Do you have this issue? Let's figure it out. We deployed resources outside of our network. So people who did not do vaccine work were enlisted to help us to develop certain aspects of the vaccine manufacturing in particular. This was particularly helpful with the lipids that we were using to encapsulate the mRNA construct. And especially with our facilities and our sterile products groups that were experts in manufacturing sterile products. As I mentioned, we adopted a risk-based decision approach. Always had a high probability of success, but there were trade-offs about when we would have data. A perfect example of that is process validation. In order to demonstrate that you have a consistent process where you're consistently and reliably and sustainably manufacturing product, you are obliged to run process validation, which means you perturb the process and then evaluate what the impact is. And you provide that data as the basis for getting approval. That was normally waived based on preliminary data that suggests that we could do that concurrently rather than as a separate exercise, which is huge. And then, as I said, we performed all of our activities in parallel. There was even, at some points, we were doing analytical development while we were doing process development. That's a dangerous thing to do because sometimes you'll get an errant result and you don't know it's the analytics or it's the process. And so you have to evaluate those simultaneously. When that did happen, and it happened in a couple of instances, we had essentially teams jumping in immediately to address that. And that's, again, one of the reasons we were able to do it very quickly. There was good communication and rapid decision-making. I will say this, it was top-down. Our CEO wanted to meet with us every single day. So there were a group of 9, well, it started out with 9 and it ended up with about 15 individuals, and that meant Saturdays and Sundays. So from June, I think it was June 15th through December 31st, we met every day at 4:30 in the afternoon. And he wanted to know, and he also pushed really hard for us to push on others to move as quickly as we could. There was good to that, but it started to get old because you can't keep that up forever. The other thing that I think is really kind of important is that the regulators were on board with helping us do that, as I said before. They were reviewing protocol information and data in real time. And that's something that we ought to think about in the future for some of these pandemic options. Is it possible to set up some sort of system where that can be done, not just with one regulatory authorities, but with all of them? Now, we also played a little bit of hardball. And the reason I say that is because there were some countries, and I'll call them out here, not because I'm trying to out them as bad countries or negative countries, but who had statutory requirements that they could not waive. And the TGA in Australia was one of those countries. They kept saying, we have to test every batch of product that comes into our country. Now, we test to release the product. They repeat those tests that we transfer to them, as does every country in the world. Vaccines are the only product where that has to, well, gene therapies and cell therapies, but before them, those are the only products that have that statutory expectation. And we basically said, look, we can't afford to do that. If you don't want the product, that's fine. You won't get it, but you're not going to test it. You're going to have to rely on another country that we're working with. And in this case, Germany was willing to do some of the testing because we had transitioned some of the methods. And our release data as the basis for releasing it in your country, you won't get the vaccine. And that seems a little bit harsh, but it was necessary to be sure that we got it to as many countries as possible, because otherwise we weren't going to get it out to the entire world. We were only going to get it out to certain countries. And to this day, we still don't have it in China. Pfizer does not manufacture, at least to our knowledge, our vaccine, the mRNA vaccine in China or for China, because there was a need there for so much data and information that we could not provide. We didn't have it. It wasn't because we had it and we were trying to hide it. We just didn't have it. That they were refusing for us to be able to do that. So that's the one country in the world where, and if you know anything about China, they've had lockdowns in many of their precincts in their country for the last year and a half because of the pandemic. So it's one of those, again, a government decided they just weren't going to do this, and in that case, they didn't get the vaccine. They have their own vaccine versions, and they felt that those were reasonable, but I think they wanted to be able to get the mRNA vaccine as well. The waivers, though, as I said, were really important. When there were waivers, it made it a lot easier to move things forward. That was getting rid of some of the bureaucracy and red tape. And there was a lot of regulatory flexibility. In this case, as I mentioned before, we were able to leverage prior knowledge, and because we made so many batches of material, and we had a fairly consistent manufacturing record, most regulatory authorities were willing to waive additional data so that they could get the vaccine sooner. And that occurred even when we provided post-approval changes, optimizations to the process, changes in the analytics, things that made the variability of the overall process less concerning for them and for their for the subjects in their countries. We streamlined a lot of the processes for batch release as a result, and our approaches really enabled for rapid improvement. We had, for the US and Europe, I believe it was over 50 post-authorization or post-approval changes just for manufacturing in the first year, which is a huge amount. But again, we were extending development into commercialization much more than we normally do. So a summary of the accomplishments that we had to date: the development, as I mentioned, fit for purpose, parallel development. The mutual reliance and recognition was huge. I can't say enough, and that is something that we're trying to get regulatory authorities now to embrace, not just for vaccines, but in general. There doesn't seem to be much of a reason, in my view, other than the need for some sovereignty in certain countries, not to accept some opportunities for mutual reliance and mutual recognition. Remember, when we manufacture something, we usually do it one way, usually in one group of places. And yet we have to go through 167 reviews of the same information. Doesn't seem particularly practical. And when regulators complain about capacity, this mutual reliance and recognition opportunity is a way to reduce that. They can rely on each other rather than having to have their own individuals. So far, we've delivered about 5.7 doses globally to about 100 countries. And probably the most promising thing from my perspective is this improved stability and storage. We're now seeing with certain versions of our vaccine, and this is the wild type vaccine. I haven't talked about the other strains yet. We're starting to see some stability that we can manage at 2 to 8 degrees for a longer period of time, and even at -20 rather than at -80, which is really going to be a boon for us as we move forward. Okay, so where are we going? Alan, I think, painted this picture, and I think Yogi Berra sums it up very nicely. The future ain't what it used to be. You've got to be very careful if you don't know where you're going, because you might not get there. That's kind of where we're at right now. There's an awful lot of thinking about different things that we'd like to do, but no decisions have been made. And we're starting to lose this collaborative effort. And with the exhaustion that we're seeing, particularly in the US, there's less appetite for vaccines. So how do we manage this going forward? In the future timeline, these are some of the things that Pfizer has been working on. And I had to kind of keep them. I couldn't share the details because a lot of it's proprietary. But we've been working on a lot of variant constructs, particularly with Omicron. We've looked at Delta and Beta as well. Delta because of the severity of disease associated with the Delta. Omicron, because it keeps changing, we're trying to figure out if there's a way to address it in different ways. Bivalent vaccines, clinical studies for generation of vaccines that are looking specifically at the structure of the spike proteins. We call it the super spike mutations. And we're also looking at the possibility of some focus on T cells. We have a lot of different ways for administration and changing the formulation. Again, we're trying to make this as robust a process and amenable as much as possible to a sustainable and reliable platform so that we can interchange constructs, different variants, different mutations as much as we'd like. And we may at some point get to a pre-filled syringe, which would be very significantly nice for moving into regions in the third world, because that is something that's much more easy to administer generally. The criteria for the next phases of development include all of these, and I've just kind of gone through those on the previous slide. One that I'll point out, though, that has been kind of interesting are these modified spike and T-cell targets that we're looking at. Again, it could be promising, it may not be, but it's something that we have to look at as an alternative to boosters and constant vaccinations. If we can find something that will actually improve the durability, we may get to a point where we're in a better place with this vaccine and with the other vaccines that may emerge or need to emerge to address the mutation of the virus as it proceeds. One of the other things that I put on the bottom is combination products. I did highlight in I think last year's presentation that we were already looking at the possibility of combining this with the flu vaccine. Now, COVID is not seasonal. And as a result, is it wise to do that? There's been a lot of debate about that. In our company, I can tell you the debate has been really heated. But because I think most of us are realizing that there is exhaustion, it behooves us to consider how we can combine some of what we're doing so that it's not as onerous on the population and it will sell better. And I don't mean for money, I mean sell the whole notion of vaccinations, because as you know, there's still a lot of reluctance. Durability is still a big issue. Alan talked about this a lot. Consistency still has to be maintained for all of the changes, and we have to maintain that not only through what we do in our manufacturing, but we actually have to run clinical trials to demonstrate lot consistency at some point. And in the US, we were able to get that waived. In Europe, we were able to get that waived. In Japan, we were not able to get that waived. And so we had to provide data to substantiate that. We had to provide some clinical data. It was real world evidence, but nevertheless, we had to collect it. We are looking at maternal safety. So in other words, when do we administer this to pregnant women? And at what point is that an appropriate approach? We did run some clinical trials, similar to the clinical trials we've done with RSV patients, because we are also looking at an RSV vaccine that's not mRNA related. Immunocompromised patients. They're the ones that we're worried about the most, obviously, because they're the ones who could be put in a position where the vaccines either cannot be administered or they don't work. Multivariant and valent adult and pediatric with and without boosters. So Alan kind of touched on this. That covers about everything right now. We don't know what this looks like. And as Alan mentioned at the VRPEC yesterday, while there was an agreement in general about proceeding with the bivalent vaccine, it was not clear which Omicron version is going to be included in that, with the wild type or with the Delta version or even Beta. Because Moderna, I know, ran vaccine studies with Delta and Omicron, but it was the original Omicron. And Pfizer ran the Omicron with the Wuhan. So, and as I mentioned, the combination products. There are also some future trends just in general for these kinds of mRNA vaccines. I mentioned the stability. The stability is always going to be a problem for this particular platform unless we can find alternative ways to manufacture. And there's some promising work being done right now at a number of companies, including ours, to try to get to that point, because mRNA as a platform is going to be extended beyond COVID. It will most likely be applied to almost every other vaccine that you can think of. Shingles, RSV, you name it. And there's a lot of reason for it because it is relatively straightforward and from a manufacturing perspective, probably one of the easier types of manufacturing approaches that we can take. The one area that we really need to learn from, this is a lessons learned that I would like to see start sooner rather than later, and I have not seen enough of this, is in the improved pandemic preparedness. I think we were abysmal when the pandemic hit in terms of our preparedness, and I mean globally. Each country had their own view of what to do. Each country wanted to do it differently, and we ended up having to play a lot of intermediary discussions with many of those countries to try to convince them that there was only one way this was going to work, if they all worked together. And they did. Are they going to continue to do that? We don't know. I do think some of the things that they agreed to do, like waivers and adding flexibility, understanding where the capacity was in order to do this effectively, and then the harmonization on expectations was tremendously beneficial, and I don't see why that can't be brought forward. And it's one of the things that we've been pushing very hard. I know Moderna has pushed it with us, particularly with trying to get the FDA in Europe to agree, because they're sort of the standard bearers for regulatory authorities around the world. But it has to be done, or we won't be able to face another pandemic, or even really get out of this one. We're still in it, whether we like to admit it or not. The one big issue that's going to confront us, and this is where the money comes in, is local manufacturing and distribution capacity. We're being asked as a company to give away our intellectual property to different countries so that they can do their own manufacturing in a modular way locally. That's not going to happen. I hate to say it, but there are commercial interests here, and they're not going to do it very easily. It's one of the few areas where I think that there's going to be some significant debate about how to do that, or how effectively to do it. If we can't even talk to each other, it's going to be harder to talk with individual countries and give them the IP to do that themselves. There are likely to be some very high bars for new versions of vaccines. Regulators are already asking us, even with the new versions, to do more in the way of safety and immunogenicity data in advance. This came out of the VRPECs that we've had so far, but it's also come out of day-to-day interactions where we've suggested we can do something relatively easy with this new construct. We'll put it in. We don't need clinical data. Nuh-uh. You're going to need some clinical data. They still want to have that. I think there's a possibility of leveraging some of the risk or lack of risk, but it's going to be difficult sell. Peter Marks made it clear that the FDA does not see adding such a variant strain only as a booster. That was part of the VRPAC yesterday. I think that's a very interesting comment. He felt that it would have to replace regular vaccinations in addition to boosters. So what does that mean? Okay, how is that going to work? If every company's got different boosters and different versions. The CDC and the FDA officials are not in favor of simply adding booster shots. That also came out of the VRPAC yesterday. Waning immunity, as Alan pointed out, poses probably the most significant concern, particularly as we're approaching the fall and winter flu season. We don't know what that's going to look like. And there's really no good framework to decide when to response to a significant or dangerous loss of protection for people in the real world. Who decides what strains to make? Alan raised this question, I'm raising it as well. Do manufacturers continue to manufacture vaccine or switch? We've got a glut of Wuhan vaccines right now. We made as many as we could, as quickly as we could, and now a lot of them are sitting and expiring. We have been able to move them to other countries, as other countries have started to approve or authorize the use, but they may even be obsolete at this point if we're based on Wuhan and we need the new variants. If the vaccine design is changed, it's got to be for all vaccines, according to Peter. This was another quote from him yesterday. And no one disagrees with that, but who's going to make that decision? What's going to look like? Is Moderna and Pfizer going to get together in a room for their mRNA vaccines and come up with the right constructs? Vaccine manufacturers need to target the same strains under FDA and VRPAC guidance. I don't think anybody disagrees with that, but the guidance hasn't been as clear as we'd like it to be. The decisions have been limited, as Alan pointed out. Paul Offit, who often has very sage things to say in these meetings, and sometimes he kind of pisses his colleagues off from time to time, he wants to look at the T cells more than the antibody levels and focus on that area, which is something we're looking at and something some of the other pharmaceutical companies are looking at. Alan's not convinced, and rightfully so, because this is an area we haven't really explored enough to understand whether or not it's got merit. And then this last comment, given our effectiveness against hospitalization in immunocompetent individuals, which is well over 80% for many of the vaccines, we may have to accept the level of protection and use other alternative ways to protect individuals with other therapies. Well, we're sort of doing that already, aren't we? With the Omicron, even though it's not putting people in the hospital as much or causing death, a lot of people are getting Omicron, the different versions, and they're getting sick and they're taking these alternative therapies. Is that the direction we want to go in as a policy? So Alan already mentioned this, so I won't go into this. You've already covered this pretty well. Oh, the second one, you mean on favorable disposition. So there was some discussion about whether or not the sublineages 4 and 5 versus 1 should be moving forward, but there was no final decision. That was just I think parenthetically saying that might be the direction to go in. But I know my company, when we got back together yesterday, we talked about whether or not we should go for 4 and 5 or wait for the next version. I mean, where do you stop? There's certainly the development of the bivalent vaccine with either a prototype in Omicron or the Wuhan, or what we call the wild type or Omicron, or one of the other versions might be the way to go. But there has to be what I would call a consistent decision across the board, which we do not have. And then the final bullet, which I think is really important, and it was something that we felt should be under consideration by the VRBPAC, by the CDC, by the FDA, is that being able to extrapolate available data for all age ranges. Now, that's a dangerous supposition because what we saw with the pediatric studies that we did not mimic what we anticipated from the adult versions. It wasn't off hugely, but it wasn't on hugely. We feel there was a dose relationship. The dose was much lower. And perhaps if it had been higher, it would have been better. The problem we have, as you know, with most vaccines, you have to balance immunogenicity with reactogenicity. And so for patients, particularly pediatric patients, if you give a very high dose, the reactogenicity can be very high, meaning higher fevers, more localized swelling, things of that nature. So it's a real balance, and it's one of that has to be addressed. So some of the opportunities emerging from the pandemic itself, there certainly were the transformational approaches that I mentioned. We can accelerate and adapt when we want to, and when we have a pandemic and there's urgency. There's got to be transparency and commitment. My company, Pfizer, I think has really high integrity. We always have. I joined the company because of that, because I believe that the scientific integrity was where they focused most of their energy. And we were very transparent with the agency beyond what we typically are. Where, we did certain things that we never felt were relevant. We gave them everything. And we allowed them to weigh in. And by doing that, we had outstanding conversations. And we're able to move forward. We also made commitments. Where they felt there were deficiencies, we were willing to commit to address those, even if we didn't do it immediately. And they were willing to give us some room to operate. We need more harmonization and alignment, and there was a harmonization alignment during that first 2 years. I never saw so many regulatory authorities working with companies as well as they did during this time. It was outstanding. Yes, there was some negotiation, some of it a little stiff-arm negotiation on our part, but nonetheless, it went in the right direction. And finally, collaboration and communication. There was some really good collaboration and communication. We can't lose that. We cannot lose that, because if we do lose that, then we're not going to be able to handle the pandemic, and we're certainly not going to be able to handle how quickly the different versions that are changing right now, the new strains, are coming online. We need to reconcile who gets priority for the vaccine as one of the challenges. How many boosters and how long? What's the seamless transition for the variants? Can we get alignment between companies? So far, Moderna and Pfizer have been competing in the last year rather than collaborating. So we went from collaboration to competition. And again, because we've started to commercialize these things. And money is the root of all evil. And I shouldn't be saying that. It's blasphemy for someone from the industry saying that, but I'm sorry. I'm more science focused than I am commercially focused. Can import-export of materials be streamlined? One of the issues we had during the pandemic was being able to get raw materials and move them between countries, because some countries weren't getting the vaccine as quickly as they wanted, so they were actually holding us hostage for certain materials and not allowing them to go to the manufacturing site so that we could manufacture a vaccine for them. How can mRNA technology be shared to increase capacity? If we did share the IP, is there a way, and I know there's got to be a way, where we could have that available to different parts of the world simultaneously, so there isn't competition between countries to get vaccine in a pandemic, and still reap some rewards that could be funneled back into doing more research and more development? And then how do we keep track of the variant vaccines? How to keep track of which variant vaccine should be administered when? So the reason I wrote that was because now we're facing an inventory that is so complicated with so many different variations that we are having trouble keeping track of what's out there and how much we've sent to which country. And then when we've sent to different countries, how much of it is actually being used or just sitting in a warehouse somewhere. That makes it difficult to plan in the chain. Example. Israel did a really comprehensive study of a number of people who had been boosted with the vaccine over time to determine what the impact of some of the data that Alan was suggesting of what the Israelis did. But they did it, and at the time they did it, there were different strains. So now you're looking at from one, it's like a snapshot in time, and with another snapshot in time where you have different strains. And can you make predictions from that? It's very difficult to draw conclusions and extrapolate when the versions of the virus are changing so frequently. Yeah, I could see in the future, ending of how this virus is mutating. You could actually start a model, use the model as to put all of the worst possible cases into the model, and then make some predictions based on that. By any stretch. The reason I say that is because I see that when I look at some of the small molecules when we look at pharmacodynamics and kinetics, you can actually build in vitro models that are going to give you a better view than the in vivo data that you generate with the variability of certain animal models or certain human models. But we're not there yet because we still don't have all the understanding and that concerted data package. People out to these indigenous tribes around the world to evaluate what you were doing. They did anthropological studies to understand what it was in the diets and the lifestyles of these people that prevented them from getting certain things that many of us got, whereas they were susceptible to things that we found, like the common cold, that could actually kill them. And understanding that has always been a huge interest for a lot of people. The question is, where's the money in it? You're going to invest in doing it because it's still theoretical in many cases, or hypothetical. So back in the early 2019, in the 1990s, as Pfizer was acquiring other companies, we ended up at one point having 170 manufacturing facilities around the world. It's extremely expensive to maintain that number. And so we started to reduce. Now, what we reduced to was 54. This is before the pandemic. And that 54 served us reasonably well because the capacity for those was essentially round the clock for the products that they produced. What we realized, though, is put us in an awfully vulnerable position, this again before the pandemic, where if one of the sites went down for whatever reason, we had an accident, or we had a material issue, or we had a quality issue, we didn't have a backup. And so we started to build or contract. We do a lot of contracting with other companies to ensure that we had that kind of backup. And they would manufacture a certain number of batches of these different products from time to time and get them on the market. With the pandemic, we realized that we can do things in smaller facilities, more modular, and in fact have moved very quickly with continuous types of manufacturing for certain of our products that can allow us to actually be portable. And in the case of mRNA, we actually think it could be portable. We haven't, it's a prototype that has not been demonstrated yet. But ultimately, we think that might be a panacea to the pandemic in the future, or any pandemic in the future, if we can get to that level where you can do it. It's kind of like having a lab on wheels, same kind of idea, having a manufacturing facility available to be able to do that.
Along that line, because a lot of the efficacy data that people presented was taken, some, where the studies are a month or 2 months or 3 months out, whereas, we certainly know there were vaccines that you and I got when we were kids that supposedly were still somewhat protected. And so, is the definition perhaps changed too much? Should we be talking about how long is a neutralizing response, antibody response? Yes, I realize that vaccines change continually, but let's say the neutralizing response, if you look at the area under the curve as a function of time, should that be a different metric than simply saying, yes, over this very short period of time we've protected this many people? And I realize for the type of pandemic that we're faced with, we wanted to protect people immediately. But now we're in this, perhaps we got ourselves in a bind where we're choosing a technology that that we're going to have to be protecting ourselves every 3 months until Pfizer gets a vaccine pill where I can take it every 3 months, or it comes once a month.
We were worried initially with the durability because we didn't know how long it was going to last. Not that there would be different strains or mutations, we knew that, but even for Wuhan, how long it would last. So when we started to get some data on the wane, and Israel again came to the fore with that, they provided a lot.
Which efforts do you think, a 3-part question, which effort do you really like that is going on right now that is being funded? Are some of the U.S. NIAID efforts that you think these pandemic preparedness are really going to make a difference? That's question 1. what would you do to really try to develop a pandemic preparedness? So it ties into those first three-part question as we wrap.
So we're reacting right now. We're not being prospective, and that's the worst place to be. But we don't have a choice because, well, at least initially we didn't have a choice because people were dying, right? I think we have to change the stratagem that we have now from reactivity to looking longer. He asked the question about being able to predict. We have to get to a point where at least we have some reasonableness in our understanding of where this might be going. That would answer all three of your questions a little bit.